Hello everyone, my name is Yvonne. I'm a product manager at LEXOGEN, and today it is my pleasure to give a short introduction to Luthor, a new technology with the potential to revolutionize single-cell sequencing. Conventional single-cell methods are limited by low efficiency and conversion rates. Therefore, they require the analysis of hundreds to thousands of cells. These cells are sequenced at a very shallow depth, and highly abundant transcripts are captured. The transcriptomic profiles of cells clustering together are then merged to an averaged expression profile for that particular population. In contrast, Luthor offers unparalleled sensitivity and thus allows an in-depth view of the transcriptomic landscape of one individual cell. Higher sequencing depth can be applied, and even medium and low abundant transcripts are captured, so that one cell can be analyzed at high resolution. First, the cells are selected. Luthor is optimized for 1 to approximately 100 cells per reaction. Cells can be either used directly from suspension, or they can be sorted into Luthor lysis buffer, for example with the fax machine. Cells can be stored at -80 degrees for 3 to 6 months for convenience. Or cells are directly lysed and Luthor libraries are prepared. How does a Luthor library prep work? The first step is THOR, a direct RNA amplification technology. THOR amplification works selectively on polyadenylated RNA and efficiently excludes ribosomal RNA, which makes up the majority of RNA molecules in a cell. The first step of THOR amplification is annealing of an oligo-DT primer to the poly-A tail of the endogenous mRNA. This oligo-DT primer already contains a partial Illumina-compatible sequence, shown in blue, and a T7 promoter sequence, shown in brown. Reverse transcription is performed to generate a cDNA molecule, shown in gray. The main function of this cDNA strand is to remove the secondary structures in the RNA and stabilize the template mRNA for amplification. The cDNA itself is not used as a template. In the next step, the poly-A tail is removed and the T7 promoter sequence that was introduced in the previous step is converted to a double-stranded and thus functional promoter. The resulting duplex consists of the original mRNA molecule, which is now fused to a functional T7 promoter, and a stabilizing cDNA strand. The cDNA strand is unbound for amplification. THOR amplification uses T7 polymerase, which can now initiate at the promoter located at the 3' end of the endogenous mRNA molecule. In vitro transcription generates antisense RNA copies, shown in light blue. The original mRNA is repeatedly copied by multiple transcription events. The major novelty is that the original RNA molecules are used as a template instead of a copy DNA intermediate, which is regularly used in conventional single-cell methods. So even if you missed an RNA molecule in the first round of amplification, you still have the chance to capture it in a later round when using Luthor. THOR amplification, which can reach more than 100-fold, results in a high concentration of antisense RNA copies. Subsequent library generation uses these amplified RNA copies as input. Luthor libraries are generated in a highly efficient one-step conversion. Random primers containing the partial sequence of the second Illumina adapter, shown in green, are used in a reverse transcription to generate the single-stranded cDNA. This resulting single-stranded cDNA library now contains both partial adapters, P5 in green and P7 in blue. The insert sequence corresponds to the 3' end of the original mRNA, and the poly-A stretch is located at the end of the insert. During the final library amplification PCR, the second strand is generated. Illumina-compatible adapters are completed for cluster generation and indices introduced at the I5 and the I7 position. The libraries are then amplified and are ready for sequencing. Luthor was benchmarked on different cell types that were FACS sorted prior to use. Human HEC293 T cells and mouse embryonic stem cells are shown as examples. In panel A, Detected genes are shown for 1 million reads per sample at a threshold of 1 count per million. In the experiment, only 8 replicates per cell type were used. Luthor offers unprecedented sensitivity, detecting 10 to 12,000 genes from single cells with 1 million reads. Panel B shows the attribution of reads to mapping classes for Luthor libraries generated from purified universal reference RNA and microglial RNA, or from single cells. Again, human HEK cells and mouse embryonic stem cells are shown here. The majority of the reads are mapped to exonic sequences. Luthor 3' mRNA-Seq therefore reliably represents the endogenous mRNA composition and efficiently excludes ribosomal RNAs. Luthor library preps are highly robust, reproducible, and generate consistent data. Panel C shows the cell-to-cell gene count correlation for 2 individual mouse embryonic stem cells. Luthor uses a robust protocol and generates consistent, high-quality data from single cells. And all of this makes Luthor suitable for challenging single-cell applications, especially when the number of cells that can be retrieved for analysis is limited and would not suffice for a conventional method relying on thousands of cells. One such challenging sample type is microglia. Microglia are the immune cells of the central nervous system, exerting neuroprotective or degenerative functions. Genotyping of microglial cells is extremely challenging due to the fragility of these cells upon isolation from post-mortem brains. Panel A shows performance data using Luthor on mouse microglia samples. The number of detected genes is shown at thresholds of 0.1 and 1 count per million, and roughly 12,500 to 14,500 genes were detected. Panel B shows the mapping class attribution for 2 replicates of microglia. A high proportion of exonic reads are detected, and Luthor efficiently excludes rRNA and intergenic sequences. In conclusion, Luthor 3' mRNA-Seq generates high-quality data from notoriously difficult material and empowers the analysis of challenging sample types. With Luthor, you can explore further and have a look at what lies beneath the surface. Luthor's 3' mRNA-Seq offers an in-depth view of single-cell transcriptomics, and thus is perfectly suited to unravel networks and regulatory processes underlying the heterogeneity between individual cells or subtypes. You can also assess the transcriptomic changes during differentiation and development, or follow disease progression on a cellular level, for example, by analyzing the nature of circulating tumor cells. You can assess cellular biomarkers, unravel regulatory networks within cells, study cell responses, for example, during immune response or inflammation. Luthor is also suitable for applications in immunobiology, such as the study of T cells, B cells, and for ultra-low input applications. For example, it is useful to assess the transcriptomic profile in microdissections. With this, I would like to thank you all for listening. And if you want to explore further and learn more about Luthor, you can just follow the link in the description below.